Hello everyone, welcome to my YouTube channel. I am Shweta Murthykrishnan pursuing my BTech in KL University. This is a very small presentation about DNA isolation and its purification. So now let's start with the basics. What is a DNA? The full form of DNA is deoxyribonucleic acid. As we all know, there are two types of nucleic acids. One is the RNA and the other one is the DNA. The RNA, which is also known as ribonucleic acid. The DNA, which is also known as the deoxyribonucleic acid as mentioned before. So let's see what is the DNA. The DNA or deoxyribonucleic acid is a molecule which is composed of two polynucleotide chains that coil around each other to form a double helical structure which carries genetic instructions for the developmental, functioning, growth and reproduction of all known organisms and many viruses. So, the basic function of a nucleic acid is to carry the genetic information from the parental organism to the future organism, that is, from the prior generation to the future generation. Now, let's see the purpose of DNA extraction. The extracted purified form of DNA can be used for many further investigations like PCR, sequencing, plotting, etc. The extracted DNA can also be used to solve many crime cases with the help of forensic sciences. With the help of the extraction of DNA, the scientists can pursue their research based on genetics. Now, let's see the different types of DNA. There are 6 types of DNA, that is prokaryotic DNA, eukaryotic DNA, viral DNA, plasmid DNA, genomic DNA, and non-genomic DNA. Now, let's see what is a DNase. DNase, which is also known as deoxyribonuclease, is an enzyme that catalyzes the hydrolytic cleavage of phosphodiester linkages in the DNA backbone, thus degrading the DNA. Deoxyribonucleases are one type of nuclease, a generic term for enzymes that are capable of hydrolyzing phosphodiester bonds that links the nucleotides. Now, let's discuss the protocol for the extraction and purification of DNA. Most of the DNA extraction protocols consist of two parts. One is a technique to lyse the cells gently and solubilize the DNA. The second one is an enzymatic or chemical method to remove contaminating proteins, RNA or macromolecules. In plants, the nucleus is protected within a nuclear membrane which is surrounded by a cell membrane and a cell wall. 4 steps are used to remove and purify the DNA from the rest of the cell. The 4 steps that are used are lysis, precipitation, washing, and resuspension. Now, let's discuss the 4 steps of the extraction and purification of DNA from the plant cells. The first step is lysis. So, what is a lysis? The lysis is the disintegration of a cell by rupture of the cell wall or cell membrane of the plant cell. In order to lysis, we need to grind the cell in liquid N2 or liquid nitrogen and use the detergent. Now, let's go with the second important step, that is precipitation. So, what is precipitation? Precipitation is the action or process of precipitating a substance from a solution. In precipitation, DNA is separated from the rest of the cellular components. Usually in a research lab, the first part of precipitation uses phenol or chloroform to remove the proteins from the DNA. The second part of research lab DNA precipitation is the addition of salts. The DNA is then precipitated from the protein in a subsequent step with isopropanol or ethanol. The DNA is then pelleted by spinning with the centrifuge and the supernatant is removed. Now let's go with the third important step, that is washing. The precipitated DNA is laden with acetate salts. It is washed with a 70% ethanol solution to remove the salts and the other water-soluble impurities, but remember, do not resuspend the DNA. Now, let's see the last and the fourth important step, that is resuspension. The clean DNA is now resuspended in a buffer to ensure stability and long-term storage. The most commonly used buffer for resuspension is the dilution of 1 into TE buffer. TE is derived from its components tris, a common pH buffer, and EDTA, a molecule that chelates cations like Mg2+. As we've completed the protocol of isolation of DNA, now let's move on with the protocol of purification of DNA. Here, the DNA is purified with the help of ion exchange chromatography. Here, first the attachment of DNA to ion exchange particles takes place. Then, the DNA is purified with the help of column chromatography. The solutions passing through the column can be collected by gravity flow or by the spin column method, in which the column is placed in a slow speed centrifuge. Now, let's see how to quantify our DNA. Usually, DNA quantification has several methods, but here we are focusing on the DNA quantification by UV spectroscopy. Usually, the DNA and RNA absorb maximally at a wavelength of 260 nanometers. Proteins absorb at 280 nanometers. The background scatter absorbs at 320 nanometers. Hence, in order to calculate the concentration of DNA, we use the certain formula, that is, absorbance at 260 nanometers minus absorbance at 320 nanometers into dilution factor into 50 μg per ml. Now, let's see how to measure the purity of DNA concentration. The DNA concentration can be accurately measured by UV absorbance spectrometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA sample. Usually, absorbance is measured at 260 nanometers at this wavelength, and absorbance of 1 corresponds to 50 μg of double-stranded DNA per ml. With a pure sample of DNA, the ratio of the absorbance at 260 nanometers and 280 nanometers, that is, absorbance at 260 by absorbance at 280, is 1.8. Hence, ratio of less than 1.8 indicates that the preparation is contaminated either with protein or with phenol. Now, let's see the applications of extraction and purification of DNA. The ability to extract DNA is of primary importance to study the genetic causes of diseases and for the development of diagnostics and drugs. It is also essential to carry out forensic science. With the help of forensic science, many crime and murder cases can be solved. It is also essential to carry out sequencing genomes, detecting the bacteria and virus in the environment, and determining the paternity. Thank you all for listening. And if you have any doubts or further queries, you can mention in the comments below. If not, you can contact me directly through my email ID, that is mkshweta2001@gmail.com. Thank you.